Hello, Assalamualaikum. Today, we will present about Alzheimer's disease. Our group members are Fairah Hanim binti Ismail, Siti Rehana binti Muhammad Zaini, Nur Syairah binti Ahmad Subhi, and the last but not least is Siti Nur Atirah binti Muhammad Kaspi.
Alzheimer's disease is an irreversible, progressive brain disorder that slowly destroys memory, thinking skill, and the ability to do simple tasks. Those with late onset type will experience this disease. The receptor that involved in this disease is G protein coupled receptor. The causes of this disease: first, alteration of gene expression, which is number of G protein coupled receptor gene change. Second, formation of plaque, which contain the amyloid B protein. And lastly, formation of tangle, composed of tau protein.
In healthy neurons, there are two natural proteins, which are tau protein and amyloid precursor protein. Tau protein is a natural protein that stabilizes microtubules. It maintains the integrity of cellular microtubules, so it will not break apart. Tau protein works together with a globular protein called tubulin, while aiding the assembly of tubulin in microtubules. This makes tau protein achieve its control of microtubule stability through isoforms and phosphorylation, but the exact binding on microtubule of tau protein is not known. The next one is amyloid precursor protein. Amyloid precursor protein is a natural protein that crosses the nerve cell membranes. It helps neuron grow and repair itself after an injury. After it gets used and broken down and recycled, it will naturally undergo regular turnovers by two enzymes, which are beta secretase and gamma secretase. This one, the amyloidogenic pathway. Beta secretase function is to cut APP from the external side, and gamma secretase is to cut APP from cytoplasmic side. Then it will produce, in healthy neurons, it will produce short fragments of 38 amino acid long peptide. Same situations occur of the new 40 amino acid fragment. Then this fragment will easily metabolize and eliminated from body without any harmful effects. It will be chopped off, and the chopped off peptide will be soluble and goes away.
Abnormal pathway, G protein coupled receptor are involved in numerous key neurotransmitter systems in the brain that are disrupted in Alzheimer's disease, which is called AD. GPCRs also directly influence amyloid cascade through modulation of alpha, beta, and gamma secretase, proteolysis of the amyloid precursor protein APP, and regulation of amyloid beta degradation. Additionally, amyloid beta has been shown to perturb or alter GPCR function. And the fourth one is the progression of Alzheimer's disease is due to involve an accumulation of beta amyloid plaque and neurofibrillary tangles in the brain. Formation of beta amyloid plaque. Protein that involved in the formation of beta amyloid plaque is first is amyloid protein precursor, second is beta secretase enzyme, and the third one is gamma secretase enzyme. Okay, this is the protein and the enzyme that involved in beta amyloid plaque formation. Okay, this is a beta secretase which cut APP at inner side of a cell membrane. And this is gamma secretase, which function to cut APP at the cytoplasmic side that release 42 amino acid peptide. If mutation occurs in gamma secretase, 42 amino acid peptide fragment will be produced, and this fragment will accumulate to form hard, sticky, and insoluble clumps, which is called amyloid clumps. This plaque triggered destruction and destruction of brain. The second one is the formation of neurofibrillary tangles. Protein that involved in this formation of neurofibrillary tangles is microtubule and tau protein. This is the microtubule and this is the tau protein. The tau protein function as to hold the microtubule structure. In Alzheimer's patient, tau protein is not normal. Tau protein is highly phosphorylated. This process makes the tau protein inactive. As the result, microtubule loses its natural integrity and starts to collapse. Other tau protein that hypophosphorylated aggregate to form neurofibrillary tangles which distribute throughout the body nerve cell.
As a conclusion, the exact cause of Alzheimer's disease is not known. Alzheimer's disease attacks nerve and brain cells as well as neurotransmitters. The destruction of this part causes clumps of protein to form around the brain cell. These clumps are known as plaque and tangles. The presence of the plaque and tangles start to destroy more connection between the brain cells, which make the condition become worse. So, these are comparison between normal age brain and Alzheimer's patient's brains. So, for Alzheimer's patient brains, the hippocampus and the cerebral cortex become shrink, and the ventricles, this part is become enlarged. Alzheimer's patient brains have bigger openings due to the deaf nerve cell in that region. So as we can see, there are different between normals and Alzheimer's brain. This part for language and memory for normals, and this part of memory and language for Alzheimer's brains. Tau undergoes chemical change, become hyperphosphorylate. It then begins to pair with other traits, creating neurofibrillary tangles and disintegrating the neuron's transport system.